Good evening and welcome to those here joining with us in person in Elebash Recital Hall and to those joining us online. I'm Josh Brumberg, President of the CUNY Graduate Center. Welcome everyone to us tonight for a discussion with Nobel Laureate Dr. Venki Ramakrishnan. This event is part of an annual series held in partnership with Cold Spring Harbor Laboratory Center for Humanities and History of Modern Biology and the BGI Laureate Archives Program. Tonight we'll hear from a world-renowned scientist about his journey of discovery. He won the Nobel Prize in 2009 for his breakthrough work mapping the structure of ribosomes, the protein factories of cells. More recently, he has devoted his molecular biology expertise to exploring and explaining the science of aging, which he writes about in his illuminating new book, Why We Die: The New Science of Aging and the Quest for Immortality. Thanks to science, many of us can live longer, healthier lives than ever before in human history. Understanding how we age is crucial for our own health and well-being and for those we care about and for the society we live in. I am grateful to Dr. Ramakrishnan for sharing his findings and his story with us, and to our own Professor David Jeruzalmi, a molecular biophysicist who has generously offered to conduct tonight's conversation. The Graduate Center, as New York City's only research-intensive public university, is committed to sharing the knowledge of our researchers and their colleagues with the community which we are embedded within. This evening's event is emblematic of the work that takes place every day here at the Graduate Center and its associated Advanced Science Research Center. We have over 1,700 faculty members and more than 3,100 graduate students engaged in research and creative endeavors with a focus on generating new knowledge that enhances society. With public programs like this one, we bring the excitement of the classroom and the lab to the stage. A word about tonight's format: Dr. Jeruzalmi and Ramakrishnan will converse until around 7:30 p.m., and then we will turn to your questions. All of you should have received an index card upon checking in tonight. We'll be collecting them around 10 after 7. If you want your question to be read, block letters, legible. Okay, this is our teachable moment of the evening. And we'll collect them, and the last 15 minutes, ending around 7:45, we'll be answering some of your questions. As they say, aging can be so challenging that we only do it once. So I'm looking forward to learning how to age well. But first, it's my pleasure to welcome Dr. Bruce Stillman, the president of Cold Spring Harbor Laboratory, who will fully introduce tonight's guests. Bruce.
Thank you very much, Josh, and it's a great pleasure once again for Cold Spring Harbor Laboratory and the Center for Humanities at Cold Spring Harbor Laboratory to host this wonderful series of highlighting some of the world's most productive and most interesting scientists, and we have one here tonight in Venki Ramakrishnan. Venki is a faculty member at the famous Laboratory of Molecular Biology in Cambridge in England. But before that, he spent a considerable amount of time in the United States. In fact, he worked at Brookhaven National Laboratory on Long Island and took a course at Cold Spring Harbor Laboratory in structural biology, which I hope you'll discuss, taught by a future Nobel laureate, and that course is actually to train scientists to become Nobel laureates. So if you want to become one, sign up. He, in addition to the work that he'll talk about tonight and the famous work on the ribosome, he became president of the Royal Society of London. That is an extremely prestigious position. The Royal Society goes back to 1660 and has his predecessors include Sir Isaac Newton. So he's in good company as being president of the Royal Society, which he was for a number of years. He's written a number of books, as you've heard. He wrote a book called The Gene Machine, which is a book along with Jim Watson's Double Helix, I think really describes accurately how science is actually done from a personal perspective, and it was a great book. And then he's recently, as you heard, written a book about aging. So we're really pleased to have Venki here. And he's going to be, the discussion is going to be led by Dr. David Jeruzalmi, who's a professor at CUNY, as you heard. David I've known for a long time because he determined the structure of some of my favorite proteins with a scientist named John Kurian, but before that he was a PhD student at Yale with Tom Steitz, who was also a Nobel laureate, and I'll hope you explain the relationship between Tom and Venki, that David did his graduate work with Tom Steitz. He has been very prolific in his understanding of the structures of proteins involved in DNA replication and DNA repair. So it is, I think you're in for a great evening, and I turn it over to David. Thank you.
Thank you, Josh, and thank you, Bruce, for introducing us. And let me also join them in welcoming you, Venki, to the City of Science and to the City University of New York. It's a great honor to be here with you to discuss your new book, Why We Die, The New Science of Aging and the Quest for Immortality. So let's get right to it. So your Nobel Prize-winning work was focused on understanding the structure of the ribosome, and the person that I did my PhD with shared the prize with you, and so it was a thrill to have that happen. But your laboratory has not worked on aging in the past, and so my question here is, what was the creative spark that led you to write about aging?
I would say there were both positive and negative motives. The positive motive was that my own field is about how the ribosome translates mRNA to make proteins. And this whole idea of protein synthesis is highly regulated. The process is highly regulated. And it's that regulation that breaks down as we age. So there are a number of factors, major pathways that affect aging that work by actually slowing down or shutting down protein synthesis. So there was a really quite direct relationship between my own field, if not my very own work, and the processes of aging. Of course, aging has many different factors. At the same time, there's been a tremendous increase in interest in aging because societies are getting old. By which I mean people are living longer, but they're not having very many children. And so your age distribution is being skewed towards older people. There are fewer and fewer young people to support this growing population of older people. And so governments are all very interested in trying to do something to keep older people healthy. And so there's been a lot of interest in aging. The other is that maybe 50 years ago, I don't know if Bruce will agree with me, but aging was considered something of a backwater in molecular biology. Top-level molecular biologists simply didn't go into aging in those days. And that's changed, and that's because our perception of aging has changed from something that's just inevitable wear and tear to actual processes that go wrong. So those were all the positive reasons. So it felt like a very interesting time to write about aging. The negative side is that every week, possibly even every day, there's a news article in some magazine or website or newspaper saying, oh, the magic cure for aging has been found. Take this pill and you're going to extend your life. And a lot of it is just pure hype. And and even scientists in the aging field, whether they are consciously aware of it or not, they tend to be heavily biased. They tend to be very optimistic. They often have companies that are involved in particular aspects of aging, or they offer services. So, there's a lot of this distortion of the field. So I thought that somebody who's not got a direct skin in the game, but who's adjacent to it, adjacent enough to be able to digest some of the literature, should maybe write a book about it to let the general public have a real understanding of what's going on, what are the prospects, and that'll also allow them to have a framework when they read something, they'll have a mental framework to understand it and put it in context. Sorry, that's a long answer, but
Thank you, though. So Why We Die is your second book. Your first book was, of course, as we heard, Gene Machine, The Race to Decipher the Secrets of the Ribosome. Both books tackle fairly complicated subjects. And so in both cases, you take very complex subjects and you break them down into bite-sized pieces for the public to digest and understand.
yeah, not version 1 though. I have
Version
I have to give my editors a lot of credit for that.
So I guess working with the editors then might be the hardest part. But what is the hardest part about explaining complex phenomena to a general public?
I think a good sort of mental framework is to assume that the person is intelligent but doesn't actually know anything, about the field. And so you have to ask, if I didn't know this jargon, how would I explain it to somebody? So it's a very different process from writing a scientific paper that's addressed to an audience of experts. And another very important lesson, and this I learned in the back and forth with the editors, is don't go into minute details that are only of interest to aficionados. Get to the point. What is the point of something, and then just explain that. I think it was a good. The fact that my editors were not scientists, and for either book, by the way, they were English majors or things like that, and so that actually was a good thing, because they would push back. If I used a lot of acronyms, of various transcription factors, all have these 3-letter, acronyms. The editor just X'd the whole page and said, "This makes no sense, and it's an alphabet soup," So I had to rewrite it. So.
And do you think, what responsibility do you think that scientists have to engage the public through their writing, especially on subjects that are important, like mortality, aging, the nature of discovery?
I think there are 2 reasons why it's important. Firstly, because all of us are worried about aging and dying, we're very vulnerable to hype. I mean, that's just the reality of human nature. And so in this particular case, I think it was important to try and be as objective as possible. But I think in general, scientists do have an obligation to communicate to the public for many reasons, not least that a lot of our funding comes from the taxpayer. So it's our responsibility to sort of tell the public, what are we doing with your taxes, okay? And if we can't make that case, there's something wrong. So I think it is important. Now, not all scientists are great communicators. Some of them are best off left in the lab to do their science. But there are others who are able to communicate, and they should be encouraged rather than discouraged, which sometimes happens. And it's particularly important today because information is becoming increasingly fragmented. And you will see all kinds of websites and sources of information that sound scientific. They use pseudoscientific jargon like negative energy, for example, referring to some mental state or something, whereas to a physicist it has a very precise meaning. But they'll use pseudoscientific jargon, so the average non-scientist finds it very difficult to distinguish between that and real science. And so if scientists don't try to occupy that stage, they'll cede it to people who are really charlatans and disinformation disseminators. So I think it is important.
Great. So let's now turn to the main subject of the book. Now, Benjamin Franklin told us that in this world, there's nothing more certain than death and taxes. We're going to talk about death and preventing it. And at the end, if you want to talk about taxes, we can do that too.
As an American living abroad, taxes are a nightmare. I'd rather not talk about it.
So I think, although you try to remove many of the technical terms or to simplify them, nevertheless, there are some terms that need to be understood properly. So some of these are, as we discuss dying and aging, are average lifespan, health span, and maximum lifespan. So what are these terms?
Yeah, so I should add life expectancy to that.
And
okay?
life expectancy.
Yeah, so lifespan and life expectancy are closely related. Life expectancy usually means life expectancy at birth. That is, when a new baby is born, how long can it expect to live? Okay, so that's life expectancy at birth. It changes. So life expectancy at 60 is different from life expectancy at birth because you've already survived to 60. Okay, and lifespan you can think of as sort of average life expectancy for the species. Maximum lifespan is how long could a member of the species possibly live? And typically, in an empirical way, it means how long do the top 10% of the population live? In other words, the longest lived 10%, what is that number? Okay, because average lifespan could be the result of many things, including environmental problems, disease, pandemics, all sorts of things. So maximum lifespan really determines the sort of ultimate ability of how long you can live for a given species.
And has the lifespan and health span, have they improved?
Oh, I should talk about health span. Health span is how many of those years are you spending healthily? And we may talk about that later because it's an interesting concept. It's much harder to define because what good health is somewhat subjective. And one thing that has happened is average life expectancy has gone up over the last 150 years. It's almost doubled. So, 150 years ago, a human being on average could expect to live to be at around 40 at the most. And now, the average life expectancy at birth is over 80. So we've almost doubled average life expectancy, but most of that has happened by reducing childhood and infant mortality, deaths in childbirth for women. So those are all things, as a result of public health, vaccines, increased food supply due to fertilizers. All of those things help us survive longer. Then we still, that improved it from 40 to about our 60s or so. But then we started dying off around 60, and that was because of diseases like heart disease, cancer, diabetes, and so on, and dementia too. And we've made quite a lot of progress. For example, we can treat diabetes. Cancer rates, although many forms of cancer are still, recalcitrant to treatment, we've made tremendous progress in cancer treatment over the last few decades. So as a result, life expectancy has been creeping up from 60s to the 80s. But remember, even in the 1500s, people were living, Michelangelo lived to be almost 90. He was 89 and something when he died. So maximum lifespan may not have actually changed. It's just that the odds of reaching that limit were very, very small in the old days.
Is it surprising that the maximum hasn't changed?
Not entirely. So, the oldest person to have ever existed, at least on record, is a French woman named Jeanne Calment, who lived to be 122 years old and died in 1997. Interestingly, until she was well over 100, she would bicycle and she smoked a cigarette and had a glass of port every day after lunch. Not something I would highly recommend for longevity, but the one thing I could get behind is she also ate 2.5 pounds of chocolate a week. So anyway, she lived to be 122, but in the 30 years since she died, No one has exceeded 120, okay? So about 110 to 120, somewhere around then, seems to be the natural limit of our species. And we can get into evolution of lifespan if you want, but that seems to be the way our species has evolved. And going beyond that will take real breakthroughs in aging. I don't think it's impossible, but I think it's extremely difficult.
Okay, let's turn to evolution. Unlike the gripping spy novels that I like to read, where major plot twists are not revealed until the end, you provide an answer for why we die in the first chapter. So why do we die?
Okay, so, in biology, most of biology is about how, because the why of biology is really because evolution selected for that, okay? So the ultimate answer in any biological question is really because of evolution. And the evolutionary theory of death, or of aging and death, is the following. If you look at species, lifespan varies by, a huge spread. You can have an insect that lives only for a day. You can have a butterfly that lives for weeks or months. But then at the other end, you can have a Galapagos tortoise that can live to be over 150 years. In fact, I don't know if I think Bruce remembers there was a Cold Spring Harbor symposium to celebrate 150 years of Darwin's book. Well, there could be a Galapagos tortoise around today that might have actually met Darwin. That's an interesting thought. So you have this huge variation. Why is there that huge variation when we're all made of the same building blocks? We're all subject to the same environment. Well, think of a mouse. Throughout evolution, species have been resource limited. I mean, we humans have overcome that only recently as a result of not just agriculture, but fertilizers and things like that. But most of our history, we too have been resource limited. And what that means is that you have to use those resources for different purposes, and there's a balance there. You can use your resources to maintain your body and therefore maintain and repair all the damage that occurs day in and day out. That takes energy and resources. Or you can use those resources to very rapidly grow and reproduce. Now, in the case of a mouse, there's no point in having a mouse that lives 40 years or 80 years, because long before that, it'll be killed in the wild by a predator, or it'll die of starvation, or any number of external causes. So that would be a wasted use of resources. So there, evolution has selected for a species that grows very rapidly and reproduces prolifically, and that's what allows its genes to survive. Because ultimately, what evolution selects for is the survival of your genes, something that biologists call fitness. It's a sort of slightly misleading term because it's not about, building up muscles in the gym, but it's about your ability to pass on your genes. Now, if you're a larger animal, there's also another problem, and that is your metabolism is slower. There's an inverse relationship between metabolism and size. And so a larger animal like a whale can live for a few hundred years, and those species have very slow metabolism, so they take a long time to mature, a long time to produce offspring, they have to find mates and so on. And so it's advantageous for those species to live longer, and they've evolved to have all kinds of mechanisms to repair their DNA and various other repair mechanisms which are different and larger in number than even we humans. Now, an interesting counterexample of this are bats. Bats are the same size or same weight approximately as a mouse. Well, many bats are. Some, like fruit bats, are bigger. But why do these bats then live 20 times as long as a mouse? And the reason is that bats can fly around, so they can escape predation more easily. They also can forage over wide areas, so they won't starve as easily. So there, it pays for evolution to select for a longer-lived species. Now, the final thing is, it's not just that, but it's even worse. Evolution will select for something that improves your fitness, namely benefits you when you're young so that you can mature and reproduce, even if those same traits cause you to age later in life. Because it doesn't care what happens to you as long as you've passed on your genes. That's not what is not selecting for the individual body. And there are many examples of that. For example, our chromosomes shorten every time a cell divides. This is called telomere loss. And it's very related to the DNA damage, response the cell has to damaged DNA. And so it was probably a mechanism to prevent cancer. Because you don't want a damaged cell to go on and then become cancerous, because that could kill the whole organism. So something that might have prevented cancer early in life, causes you to age later in life. So that's one example.
I think you mentioned telomeres, so maybe you could say what those are.
Yeah, so if you look at our genome, it's, we have 23 pairs of chromosomes, and each chromosome is a long piece of DNA. So it has ends. Now, during life, damage occurs to DNA, and one of the more serious forms of damage is when the DNA essentially breaks off. Both strands of the DNA break off, and then the cell has to figure out how to join them again, and that's a complicated process. Now, if you have a long piece of DNA, how does a cell know that the end of the DNA isn't a broken piece of DNA? So how does it know that's not a break in the DNA, but it's actually the natural end? Well, the way it knows is that there are special sequences that are added on to the end of the chromosome, and these are called telomeric sequences, and then the whole thing is kind of organized in a special structure called the telomere with the help of proteins that, Titia DeLange at Rockefeller has done a lot of work on that in that area. So each time your cell divides, these telomeres become shorter because the mechanism to copy the DNA isn't able to copy the very ends of the DNA. So there are problems with that. And so in normal cells, the chromosomes keep getting shorter and shorter, and at some point the end unravels, and then the cell sees it as if it was a damaged DNA and sends that cell into a state where it can't divide anymore, and it's called a senescent cell. And it's worse than that. It secretes inflammatory molecules so that the immune system can come and clear it. Now, early in life, these senescent cells have a real purpose. Later in life, we accumulate too many of them, and they cause a serious problem. But that's the relationship between telomeres and aging.
I think in your book also, you mentioned that aging, which is why we are not able to exceed the maximum lifespan, that it's a progressive accumulation of damage. Is there a way to know, like for example, my watch will tell me that I'm 2 years younger than I'm supposed to be. Are there measures of aging that can be objectively applied?
Yeah, I mean, one thing that has plagued the community is how do you measure aging? And because you can't really study something that you can't measure. And the oldest way to measure aging was to ask you your birth date and calculate how old you are. But anyone who's been to a high school reunion knows that that's not the whole story. You go to your class reunion and some people are unchanged and others are unrecognizable. And so we know that we age at different rates, and how do you measure that? And people have come up with various kind of biological clocks. One of them is that as time passes and as we age, there are mechanisms to add chemical tags called methyl groups to our DNA. This alters the pattern of which genes we express, which genes on the DNA actually get, that information gets turned into proteins that are actually useful to, do something. So that whole field is called epigenetics because it's a layer on top of genes, on top of genetics. And so you can look at these methylation patterns, and it turns out that by some criteria, looking at the methylation patterns gives you a more accurate picture of how old a person is than calculating the number of years since their date of birth. And so it was thought to be a proxy for biological age. Now, the trouble is there are lots of methylation clocks, and and people will gladly relieve you of a few $100s to tell you your biological age by taking a sample of your DNA and looking at your methylation patterns. And if you go to 6 different companies, you'll get 6 different answers, okay? So that is one problem. The other problem is there are other ways of measuring biological age. One of them is to look at the distribution of all the proteins in various tissues. This is the so-called proteome, which is the collection of all your proteins. And that changes with age. So that's another measure of age. Another is, as we get older, sugar groups get randomly added to our proteins. And they also cause, a failure of function in some proteins. But by measuring the level at which sugar groups are added, you can calculate somebody's age. Just like when you go get tested for diabetes, they're looking for the addition of a sugar group to a protein in your red blood cells called hemoglobin. And that's a test for whether you've got elevated sugar in your blood for a long time and therefore might be diabetic. But this can happen to all proteins, and as we age, that's a problem. And so there are all these clocks, and one of the things that I did suggest in my book, and this is one problem with being an outsider writing the book, is I said, well, these people should actually get together and compare their clocks and agree on, some, not just one clock, but maybe a set of clocks to monitor aging. But actually, the aging community actually has done that. I mean, the aging research community actually has done that. They get together, and there's a sort of clock consortium where they compare clocks and so on.
So the whole story about aging is that our body is accumulating damage. Don't we have ways of counteracting that damage? I know DNA is well known to undergo damage just in water, yeah. and we have repair mechanisms. Why don't those work?
Yeah, no, it's absolutely true that our DNA gets damaged all the time. There are thousands of events in every cell in every day. So. And it happens even without any environmental factors like, it's not about, dangerous radiation or environmental carcinogens or anything. Even if you had it in very, very pure, protected conditions, you would still get damage. And so very early on, even in the earliest forms of life, like bacteria, there are mechanisms to constantly survey the DNA and repair the damage. However, the cell also senses if the level of damage is too high to repair. So then, what do you do with a cell like that? If you will let it carry on living, it could be a cancer risk. And a single cell that becomes cancerous in an organism is a threat to the entire organism. But a single cell, if it's killed off, has no consequence whatsoever, because we're getting rid of millions of cells all the time as we live, and that's just part of life. And so the cell has evolved mechanisms to send these cells into that state called senescence that I told you, or it can, in extreme cases, it can send the cell into a form of suicide called apoptosis. And exactly why it chooses one versus the other is still, not entirely clear. But, it has mechanisms to deal with it. But the result is aging, because you're creating more senescent cells and so on, simply because you can't keep up with the damage. And at higher levels, it's the same. We have ways, for example, proteins are made that are of poor quality, and we have ways of dealing with poor quality proteins, or proteins that get unfolded and start clumping together. There are all sorts of mechanisms to get rid of these proteins and recycle them. But as we get older, we can't keep up because the rate of these aberrant events becomes larger than our ability to keep up with them, and that's one of the reasons.
So you've mentioned senescent cells a few times. I read somewhere and in your book that there are efforts to try to discover these cells and to eliminate them, and that could be a way to fight aging. What is the status of that?
The idea is that as we get older, the number of senescent cells becomes too large for the system to deal with in a normal way. And so if we were to, and also these cells cause inflammation causes more organ damage and more, causes more senescent cells. So it's a vicious cycle. And chronic inflammation is generally one of the major problems as we age. And so one idea is that if we were to kill off senescent cells, that should improve health. And indeed, by targeting senescent cells have some characteristic processes that are turned on inside them. And so if you can, specifically recognize those processes and target them, you might be targeting senescent cells specifically. And people have tried this in mice and shown that the older mice then appear healthier. They appear to be generally younger in terms of by various criteria. And so this has led to a big boom in this field called senolytics. So people are looking for drugs that will hit senescent cells, and some of these are in clinical trials. Now I should point out though, we have senescent cells right from early embryonic stage. They're actually important in development. They're now thought to be important in development, in pattern development and various other things, just like cell death, which was thought to be a random process. Now we know that there's a thing called programmed cell death, where cells die at a particular point in development in order for the organism to develop normally. And so senescent cells play a very important role in biology, and they probably continue to have important roles, even when we're older. So if we were to kill off all your senescent cells, it probably wouldn't be a good thing. I mean, nobody's tested it, but we know because they have fundamental biological roles that it is unlikely to be a good thing. But the question is, can you just restore the balance so that you don't have too many senescent cells? And that's the goal of the industry.
Okay, I just want to also just take a moment to ask the audience to write down any questions that you might have on your cards, and someone will come and collect the questions. And as has been mentioned, the larger the font size, the better. Early on, you mentioned that some animals live longer than we do, and some of them also, I read, seem to age much slower than we do. Is there anything that we can learn about how they do it that could be applied to human lifespan?
I think, there are people who study particularly long-lived animals. One favorite of the anti-aging community was this very ugly rodent called the naked mole rat, which is, I think, found in East Africa and lives in these burrows, and lives many times longer than what you would expect for its size. But if you look at these animals, there are a number of things that emerge. Most of them have slower metabolism, and but of course not things like bats, which have reasonably high metabolism. Another is that they tend to have more repair mechanisms. That is, we all have repair mechanisms, but they have more of them, either in quantity or in the number of types of repair mechanisms. Whether we can directly use that to help ourselves is not entirely clear, okay? I think they're interesting to study in their own right, and a lot of interesting biology will come out of it. But in terms of pure application, will that improve our health in old age? I think only indirectly, in that we might learn what factors are important for longevity. But of course, we can't really have a much slower metabolism and still remain humans, and so without changing our, intrinsic makeup, I think it'll be hard. But what we can learn are general rules of biology of what causes aging, and that can be useful to us.
Okay. Earlier you said that 50 years ago studying aging was kind of the backwater of science, and now it's a very prominent area. Some of that may be because the scientists have aged. And in your book, you also suggest that there are almost 1,000 companies focused on the problem of aging and increasing lifespan.
And tens of billions of dollars.
And tens of billions of dollars, yes. Why do you think that living forever or living longer has such a deep hold on us humans?
I think, there are two things to this. First of all, if you talk to many of the scientists involved in longevity research, except for a few, what I would call on the extreme, life extension wing of the population, most of them will say, we're not trying to extend life, we're trying to extend health. Of course, I'm a little skeptical about whether you can extend health without extending life. I think if you were to extend health by actually tackling aging, there's an equal chance that you'll also extend life in the process. And so you might still have that slow period of decline, except that you've postponed it. Instead of happening between 70 and 85, maybe it'll happen between 85 and 100 or something like that. Okay, so that's one thing. The other is there are people who want to extend life. They say, why should we die? It's kind of a shame. We're here, we're alive, we're thinking, conscious individuals, and we shouldn't. There are people who just one billionaire, tech billionaire, Brian Johnson, has a T-shirt which says "Don't die," and so. But that's human nature. If you look, it goes back, centuries. The first emperor of China was somebody named Qin, which is probably why it's called China. He was obsessed with longevity. He would try all kinds of potions, and he would send people off to look for elixirs of life. And it may have killed him off because he probably took all these compounds that had mercury and shortened his life, paradoxically. And he died in his, when he was about 40. So that's been true all along. And even today, if someone were to come to you and say, look, we've developed a pill, we know it's perfectly safe, and it's going to give you 10 years of extra healthy life. I mean, the key is healthy, obviously. So most people would take it. I'm actually taking 2 such pills at the moment. Well, actually, 2 combinations of such pills. I'm taking a statin for my cholesterol, and I'm taking high blood pressure medicines. Now, you wouldn't think of these as anti-aging medicines, but actually, their goal is to keep me healthy for longer and possibly extend my life, so I don't die of a stroke or a heart attack, sooner rather than later. And so I think that's just human nature.
You mentioned the anti-aging interventions, and some of them are, get a lot of media attention. Maybe we can go through some of them to see how much they're grounded in solid science. And some of them you discuss in the book. So let me list a few, and then you can discuss. So caloric restriction, rapamycin, metformin, and then NAD.
Okay, so caloric restriction is based on the idea that, animals, if you restrict the number of calories they consume, so they don't starve, they're not losing weight, they're stable, but they're getting the bare minimum of calories and any nutrients they need, like vitamins and protein and so on. And these animals tend to resemble younger animals, when they're old. And they also extend lifespan by about 30% or so. So, 30% would mean, if we're 85, 30% of that is significant. It's about 20 more years, right? So there are people who advocate caloric restriction, and there are a few people who practice it. But I can tell you it's extremely hard. The animals who are on caloric restriction, the moment you take them off that diet, they start gorging. as much as they can eat. They're hungry all the time. They feel cold. They have a loss of libido. So it reminds me of the doctor who says, do these things and you'll live a lot longer. And the patient says, really? Doc, if I just do these things, will I actually live 10 years longer? And the doc says, well, I'm not sure, but it'll feel like it. So anyway, so that's caloric restriction. But the alternative to that is to ask, what does caloric restriction do? And we have a pretty good idea of some of the major things that happen in our cells as a result of caloric restriction. And then you ask, can you find drugs that have the same effect as caloric restriction so that you can pop a pill and then still have your blueberry pie and ice cream? And one of those pills is rapamycin. So rapamycin inhibits a major pathway called the TOR pathway, which is sort of related to my old field because it shuts down, it prevents ribosomes from starting making protein on mRNA, but that's a different story. But anyway, rapamycin was discovered completely by accident as an antifungal compound. Then it turned out to be thought of as maybe an anti-cancer compound, and then people found out that actually it was an immunosuppressor, so it was approved as a drug to give organ transplant recipients to prevent rejection of a foreign organ inside them. And now the idea of giving an immunosuppressive drug for prolonged periods to somebody so they won't age, seems slightly problematic to me because they'll be more prone to infections. They have all various other side effects, like if they get injured, they're more slow to heal the wound because that requires protein synthesis. So I think it's problematic, but the advocates of rapamycin are very gung-ho because of animal trials, and they say, well, we can adjust the dose down so that we don't have these bad side effects, but we'll still get the benefit of rapamycin. And there are some studies now indicating that that might be possible, but I still think it's one of those cases where you need very long-term studies. Now, what was the other thing you mentioned? Metformin. Yeah, so metformin is a somewhat different category because it's given to millions of people already for type 2 diabetes. So we know that it's a relatively safe drug, but it does have side effects. It is at some level, everything is, whether something is a toxin or not depends on the dose, okay? Even water is a toxin if you drink too much of it. So the point is that can you use metformin to extend healthy life? This came from an early study that diabetics on metformin were living longer than non-diabetics, which was actually counterintuitive because diabetes is a risk factor in aging. And so that led to a lot of interest in metformin, but other studies didn't quite support, in fact, came up with almost the opposite conclusion. So the studies on metformin and their effect on health span is somewhat mixed, okay? And proper controlled trials need to be done. And in fact, Nir Barzilai, who's at Einstein, is trying to initiate a human trial on healthy humans to take metformin and monitoring them over a period. And this brings to mind a general problem with aging therapeutics. If you have cancer, you'll be willing to take really nasty, horrible drugs with horrible side effects, highly toxic drugs, because the alternative is you'll die, okay? But an anti-aging therapeutic is different. You're asking somebody to take it for 10 or 20 years in the hope that at the very end they'll get another 10 years of healthy life, okay? And that's a different proposition. So I think the bar for anti-aging therapeutics is different from, serious life-threatening disease.
So in that same vein, there's currently an X Prize competition to revolutionize how we approach human lifespan. So they're offering $101 million for a proactive, accessible therapeutic that reduces the risk of chronic age-related diseases increases human lifespan, and extends the quality of life well into our later years. Now, since you will be living to 120, is there a particular moonshot approach in aging research that you see or you might be interested in pursuing in the next decades?
Yeah, well, no, I'm not going to pursue anything, but well, there are as many approaches. You talked about caloric restriction. That's resulted in a whole class of drugs. Like I would consider metformin roughly in that category, even though it's not directly related to caloric restriction. Another one of these GLP-1 drugs, which were originally for morbidly obese people or people with really severe weight-related diabetes, but now they're finding that they have generally positive effects on all kinds of things, including aging of the central nervous system. And so that's a possibility, but they also come with downsides. They lead to loss of muscle mass, and that leads to frailty, and that's a big problem as you age. So again, these are problems. I would say the one area that I think is very exciting but also very challenging is this idea of reprogramming cells. And what I mean by that is when you have a fertilized egg, there's a program that's set in place that results in the fertilized egg differentiating into an embryo, growing into an embryo. Each of those cells in the early embryo has the ability to form any of the tissues in the body. But as the embryo develops, it differentiates further into specialized so-called stem cells, which can then only give rise to certain classes of cells. Like some cells can give rise to all of the cells of the blood system, including our immune system and red blood cells. Others can form all the cells in our nervous system. So it becomes more and more specialized. Now, when you get to the final stage where you have fully differentiated tissue, you have skin and cartilage and bones and hair and organs and blood and so on, they can't go backwards. You can't go from there back to a fertilized egg. Okay, so it's essentially a unidirectional process. But it turned out you can actually go backwards. So John Gurdon, who unfortunately died at 92 just last week, showed when he was very young, I think he was just a graduate student, he showed that you could take an adult cell, remove the nucleus containing all its genes, and put it in an egg from which the nucleus had been removed, and that would grow into a clone of the original animal from where you took the skin cell, okay? It was the first cloned experiment, and eventually they did it in mammals, and you got Dolly the sheep. That shows that you can take that final skin cell and have it go all the way back and develop into a whole new animal. That's essentially a genetic copy of the original animal. It's a clone. And then 50 years later, a Japanese scientist named Yamanaka showed that if you introduced genes for 4 factors, these are factors that affect other genes. They can turn on or off other genes, called transcription factors. So if you take 4 genes, particular genes, and introduce them into a cell, you somehow start rewiring the cell so it starts going backwards. And of course, if you did this to every cell in the body, we'd all end up, with a horrible mess. Everything would be like a starting cell. You'd lose all the identity of the tissues. And so you wouldn't want to do that. But the question is, can you turn on these factors for just a short while so that our tissue sort of goes reverse in development for just a little bit, and just short enough that they're still maintaining their identity. So a particular skin cell stays a skin cell, or a hair cell, follicle cell stays a hair follicle cell. And I would have thought this was an outlandish idea, but people have tried this in mice, and they've shown that these mice looked healthier by many criteria, including those clocks we talked about. And so there's a whole community now trying to use this kind of reprogramming to see if they can use that to maybe reverse some of the effects of aging. I think it's a long way from these animal trials simply because of the way it was switched on and the cancer risks associated with it and so on. It's still a long way before you can use it in humans. But given the amount of money and the quality of scientists that have gone into this field, I wouldn't be surprised if they made some sort of breakthrough in the next decade or so.
Now, there's ample data that suggests that human lifespan is affected by social class. So as we think about these interventions that are going to increase our health span and lifespan and perhaps our maximum lifespan, how do we think about this inequality in social class and lifespan? And how do we ensure that the benefits accrue to all?
So I want to talk about two things. One is the disparity, and the second is, what would it do to society if we all started living to be 100 or 120? And that's being modest by, I mean, some of these people claim that the first person to live to be 150 has already been born, and one person at the extreme end, a British scientist who now lives in California with a very long beard, says that the first person to live to be 1,000 has already been born. Okay. So disparity first. If you look at the top 10% of the population in Britain economically, the richest 10% versus the poorest 10%, the life expectancy difference is 10 years. So the richest 10% are living 10 years on average more than the poorest 10%. That's in a country with a national health service. In America, it's 15 years, okay? And if you look at the fraction of years that they spend in good health, that number is almost double. So that means the poor are not only living shorter lives, but they're spending more of it in poor health, okay? So that's a big disparity. I should point out, my stepdaughter who works in public health pointed out that in New York City, the difference in life expectancy between the richest and poorest zip codes is 35 years, okay? And now, not all of this is due to aging. A lot of it is due to crime, poor medical care, poor nutrition, there's a whole series of external causes, but a good part of it is also due to aging. And so that's the reality today. Now, if you have sophisticated interventions like stem cells that I was telling you about, this is not going to be as easy necessarily as popping a pill like a statin. I mean, maybe it'll come to that, but not at first. And so who's going to avail themselves of these therapeutics first? It's going to be the people who can afford it, okay? And so you're going to increase that disparity even more. And that's going to create real social issues. Now, there's a social issue anyway, because as you get older, you accumulate a number of things. You accumulate wealth, you accumulate power and influence, all of which are interconnected. And so if you have a society, we already have a society where people are living longer, but fertility rates are dropping, so you're not having as many young people in the population. So you're having a society where turnover is extremely slow, which means the same people who have power are just sort of hanging on. And we see this in, I'm sure in your university as well as mine, you see, especially in academia, you see these people hanging on, running big labs and showing no signs of stopping, okay? And that's just an issue, And so there's a question of intergenerational fairness. The other problem is, I think creativity is something that, is optimal when we're young. And it's not simply because we're biologically young. It's because when we're young, we're facing the world fresh for the first time. We're looking at things new. We've not accumulated a lifetime of prejudices and dogma and so on. And so we're more open-minded. And that's part of being creative, is being receptive to new ideas. And this is why when you look at science, people continue to do good work, later in life. But if you ask, when they die, what are they going to be remembered for, okay? And if you ask that, you'll find that most of their big breakthroughs was much earlier in their lives. And it's not just science and math. Kazuo Ishiguro said in a public meeting, a public interview, but also told me that it's true in literature. If you look at authors and ask, what was their big famous work, like War and Peace by Tolstoy, it was done when they were, typically under 40. So I think, this life extension is going to create all sorts of social issues that I haven't thought about.
Okay, let's see. So many folks who heard that I was hosting you today asked me to make sure that I ask you about some things and to separate fact from fiction. There's probably too many for us to go through, but let me just mention a few of them and then you can comment. So the Mediterranean diet, the yogurt diet. I also learned that there are blue zones all over the world where there's a high concentration of centenarians, Loma Linda, California being the one in the United States. Let's start there.
I'll talk about the blue zones first because I'm really glad that I didn't write about them in my book, okay? Because I might have been tempted to somehow explain blue zones. But the reality is a paper was published about a year and a half ago, which analyzed, remember, you're talking about people who live to be over 100. So you're looking at record keeping in all of these places from 100 years ago, okay? And when you look at how accurate the records were and throw out all the ones where there was uncertainty in the age, the effect goes away. And so this person actually got the Ig Nobel Award, which Harvard gives out, for interesting kind of quirky science. But I will tell you, one thing that made me skeptical about blue zones is they were so different. You had people in Sardinia in Italy, or Loma Linda in California, or Okinawa in Japan, and they all were different genetically, they were different in their lifestyle and diet and all sorts of things. So I'm very skeptical about that. On the other hand, we know from caloric restriction studies that a healthy diet is important. So it could be that the Mediterranean diet is a balanced diet, which has got a rich mixture of fresh fruits and vegetables and fiber and things like that, and presumably they don't overeat. And so maybe it's that combination. Italy, by the way, is at the vanguard of aging. So it's in terms of an aging population, it's ahead of the rest of Europe, ahead of the US. But it's sort of like South Korea in terms of people living very long and not having very many children.
And then there's the red wine and resveratrol connection that...
Yeah, so I have to be very careful what I say here because what I wrote in the book was carefully vetted by 2 lawyers. So, but I should say, this came out of discoveries made in Boston, and but later studies have really cast doubt on whether resveratrol actually has an effect on these proteins called sirtuins, which are thought to, promote longevity. And then the question of whether sirtuins actually promote longevity has also been questioned. And so this thing has turned out to be a house of cards that's, most aging researchers are quite skeptical of it. And by the way, the amount of red wine you would have to consume to equal the amount of resveratrol in these studies would be like hundreds of bottles, okay? And so it's not that the French are getting, living longer because of their red wine.
Okay, let's turn to a few questions from the audience. So there's a question that asks if you could please say more about your journey from physics to biology to chemistry.
Oh, well, the last part isn't true, actually. I like to joke that ever since they gave the chemistry Nobel Prize to Rutherford, which was in 1908, it's been known that you don't need to know any chemistry to get the Nobel Prize in chemistry, So, no, but I did go from being a physicist to a molecular biologist. I think part of the reason was that I was working on a problem that I was not interested in. I didn't feel I had any aptitude for it. And I was reading popular science articles, mainly in Scientific American. This was in the 70s. and I realized that biology was still undergoing a huge revolution, and it's still going on, by the way. And at that time, DNA was being sequenced for the first time. We were getting our glimpse at what biological molecules look like, what membranes look like, what the immune system, the basis of how the immune system worked, or nerve transmission and things like that. So everything seemed like, incredibly exciting. So I decided to sort of punt and, just, bail out of physics and start graduate school again.
Okay, let's see. So in cancer, one of the hallmarks of cancer is that the cells begin to divide and they live without inhibition. And the question here is, can we harness the underlying process in a good way?
Yeah, people, when telomerase was discovered, there were sort of two camps. One was that, if we could inhibit telomerase, then, these cancer cells wouldn't be able to divide indefinitely, and we'd have a sort of universal, target for all cancers. And although telomerase was discovered some decades ago, that's not been, one of the more promising avenues. I mean, some of the biggest therapies in cancer have not come out of that. The other attitude was, if telomere loss is a problem with aging, maybe we can, reactivate telomerase in these cells and extend these telomeres and, thereby help with aging. And there are a number of scientific labs and also many companies that are formed with precisely that idea. But if you talk to, world-famous telomere experts like Titia Delange, just, uptown from here, she will tell you that, telomere biology is an intricate balance between aging and cancer, and tinkering with it could dramatically also increase the risk of cancer. And by the way, the person who lived to be 117 was a record holder who just died from a couple of years ago from Spain, from I think Barcelona. She wanted everything about her studied. And so the biomedical scientists actually analyzed all of her tissues and her DNA and her methylation patterns and all sorts of things, and published an article in Cell just 2 or 3 weeks ago. And she had shorter telomeres than usual. So, that doesn't go well with the theory.
Right. Let's see. So one member of the audience notes that the quest for immortality comes at the cost of increasing age and related diseases like Alzheimer's, which is incurable yet. So how do we deal with the trade-offs?
Yeah. So currently that is a big problem because people are living longer because we're tackling some diseases more successfully than others. For example, we're tackling diabetes reasonably successfully, made tremendous progress with cancer and also with heart disease. But where we've been less successful is with dementia. So as these people are living longer, they're at increased risk for dementia, which is another of the diseases of aging. And so, you could argue, what's the point of, living longer if we're all going to, get Alzheimer's at the end? That's not a great way to sort of end it all. And, but of course, one thing you could argue is that the processes of aging, the fundamental processes of aging, are similar in all tissue types. And so if we were to slow down or tackle aging itself, you would also possibly postpone diseases like dementia. So that's the goal. But as I pointed out, there's no real solid evidence that we'll be able to extend health without also extending life. So we may simply be postponing all these things to later in life.
There's a question about ribosome populations in various cancer tissues. And so there's an interest in how do epigenetic changes affect the ribosome populations in disease?
Even though I'm a ribosomologist, I'm afraid that's not something I've looked into. Okay, it is true that cancer cells, need to divide, so they need more ribosomes, they need to make lots of protein, but I don't formally know the connection.
And we have one last question. This is a great question because it's written in big block letters. So the question is what is the one piece of advice that you would have for us to achieve healthy aging?
All right, so, in my book, I end with the idea that, look, there are interventions that we can do that currently work better than any anti-aging therapy out on the market, like buying, many of these supplements. And those are primarily this trio of diet, exercise, and sleep. Now, people have been touting this for a long time, probably maybe even for centuries, but what we know now is how they work. We know what exercise does to, the biology of our cells, to its regenerative capacity, to mitochondrial regeneration. So we know a lot of things that about what exercise does. Similarly, diet is related to those caloric restriction pathways. And so we know why diet, can have an effect on aging. And sleep, it turns out, isn't just, a sort of dormant state. Lots of things happen in our sleep cycle, like repair and recycling of our components. And so it turns out it's an incredibly important aspect. And in fact, all species, even unicellular organisms, effectively sleep. They go through a daily cycle. And so those are three things, and they help each other. For example, if you exercise, you're more likely to be relaxed and you're likely to sleep better. And if you're relaxed and you're less likely to eat, binge eat and so on. So that they sort of help each other. They also reduce stress, and stress is a driver of aging because of various, direct and indirect mechanisms. And so it has broad effects that are beneficial. Now, having said that, there are two problems with it. And one is, these things also help with high blood pressure and cholesterol. So why am I on these pills? And it's because if I just tried to do it naturally, my blood pressure and cholesterol still kept creeping up. So eventually I needed to take the pills. So you could say the goal of therapeutics is maybe to go beyond what you can do yourself. But I think what you can do yourself should always be the first line of attack.
Okay, so it's diet, sleep, and exercise. That's the secret.
There are two social things which are also correlated, which I found out in the course of writing the book. One is that people who are socially not isolated, they have a circle of friends and family, they tend to age more healthily and also have longer lives, so lower mortality. The other is, and this is of particular interest to me because I'm actually going to retire at the end of this month, and that is having a sense of purpose lowers your mortality, independent of the social networking. And so I think and that could take many forms, public service, volunteering, maintaining interests, something that'll get you out of bed every morning because you want to do something. So I think those are some additional factors.
Good advice. So with that, I think we've come to the end of our conversation. So I'd like to thank you, Venki Ramakrishnan, for...
Thank you for having me.
And thank you to the CUNY Graduate Center and the Cold Spring Harbor Lab for sponsoring this lecture.